Good morning, good afternoon, or good evening, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Sam Moulton from the Business Review, and I'll be your host for today. It's our pleasure to have David Visa Clinical Research with us, who will be discussing an alternative to GMP Phase 1 trial supplies. Can you do that? Today's guest speakers are Harry Alcorn, Early Clinical Scientific Officer from DeVita Clinical Research, and John Colling, Partner from Alpha Vector. I would like to take this opportunity to remind the audience that you can submit questions using the Q&A tool, which is on the right-hand side of your screen, and you can do this at any point throughout the presentation. Please send all your questions to all panelists so we can all see them. We'll then allocate 10 or 15 minutes at the end to try to answer any questions or thoughts. So without further ado, please allow me to welcome Harry and John.
Hello, Harry Alcorn here. I'd like to thank all of you for taking the time out of your busy schedules today for this discussion on batch IP compounding as an alternative to GMP for early phase clinical trials.
Yes, good morning. Thank you, Harry. This is John Colling, so that everyone gets a distinction between our voices. And again, would like to thank all the attendees participating in today's webinar.
As we move through the slides here, we want to give an overview and an objective of what we will be talking about for the next 40 minutes approximately, and the potential benefits associated with having an on-site research pharmacy allows you the alternatives as it relates to meeting the GMP objectives, specifically looking at USP 797 with sterility, stability, assay, and good documentation to have a decreased time to make that decision for go, no-go in next steps for your IP product.
And with that, Harry, we'll also be discussing with the panel, with the audience today, those types of certifications, processes, documentations, and procedures that an on-site research pharmacy would have in place to support the bulk compounding of investigational product under USP 797 and other auspices, as well as those sorts of specific personnel training and records of training, et cetera, that would be used to support and to certify and to ensure that the investigational product that is compounded for those early phase clinical studies meet the quality expectations of the sponsor, the on-site research pharmacy, the CRO, as well as ensuring that they meet the commitments put forth in their investigational new drug application.
Thank you, John. And basically, to frame the conversation for us today and moving forward here is we all may be familiar with New England clinical compounding, and that was in 2012 where the FDA issued a 483 specifically referring to airborne pathogens, the cleaning, and the validation associated with New England compounding in regards to the issues that arose with the contamination of product being dispensed. And basically, there has been legislation that has followed this 483 from 2012, and that has addressed the 503B under the FDA guidance, as well as the state boards of pharmacy looking at this at how best do they address this gap between what the FDA oversight has and what the Board of Pharmacy has, because typically today the standard is for a clinical research site in early clinical, they do not need to be licensed by the State Board of Pharmacy. They can operate under the FDA, and they can operate as a compounding pharmacy under the FDA as well. The USP, as we all know, is a guidance, and is a guidance that is clearly embraced internationally. The boards of pharmacy now are looking at it state by state. Certain states have adopted this as a state regulation. Others are currently reviewing that as we move forward. 797 addresses the room design, the airflow, particulate count, surface sampling, air sampling, validation of the staff through process validation, and they usually look to this to be a third party, and it's not your own oversight that does occur with this type of design and certification or validation respectively in being compliant and meeting the USP 797 requirements, not only for the room design, but as well as for the staff.
So, Harry, I would also like to add for our audience members that today's presentation, while mostly focused on 797, which specifically addresses the preparation and compounding of sterile investigational product, can also be generally applied to 795 for the compounding and preparation of non-sterile investigational product as well.
Excellent. As we look at the path to considering the alternative to GMP as it relates to bulk IP compounding, we look at the on-site pharmacy that's meeting USP 797. We look at the pharmacy's experience as it relates to the staff directly. What type of testing is currently being done in the pharmacy as it relates to the environment and also to the process validation to ensure the integrity of product being compounded. And that also looking at this under pharmacy regulations as it relates to single use of products, utilizing manufactured products, i.e. Abbott, Baxter, with diluents and vehicles that are for single use only. And that if there is to be sterility using a 0.22 micron filter, and you can use a bubble point test or other tests that can be used as well. So it's a GMP-like where there's a batch record of control for understanding of the NDCs, the lot numbers, the expiration dates, whereby everything is documented and tracked as it relates to the production or compounding of the bulk IP. Okay, next slide. The batch record is really at the heart of what we're talking about in utilizing an on-site pharmacy to best understand how to move forward with the alternative to the GMP. As we look at the batch record design, the supplies that will be used, the products that will be used in meeting good documentation, but it also looks at the design, the feasibility of the site, the IP of the protocol to ensure that you not only have looked at what will be used, how it will be used, and operationally how that will occur. And all of that is done in developing the batch record with the sponsor on a clear delineation of specific activities, timed events, what is measured, what is weighed, how it is documented, and also understanding that there is a notation that will be required should there be any unusual occurrences or deviations that should occur that must be documented and best understood in moving forward after the batch record design. The demo batch is truly at the point where the sponsor and the on-site pharmacy have come to an agreement that the design, the product, the supplies, the process truly meets the intent of what we need to do to be in GMP-like, that it truly can be reproducible, it has process capabilities understood, there is technical transfer understood, and there's site-specific adjustments that will be made during the demo batch that can be noted and documented and then obviously fed back to the sponsor and the site upon review of the demo batch and results to best understand how that should be modified. This is all to assure the success in producing bulk compounded IP. So obviously we need to look at addressing sterility, stability, also the assay of the drug concentration.
And Harry, I'd like to add in this particular situation the importance of the demo batch, as you've mentioned, for our audience members in regards to really addressing site-specific needs and processes for the investigational IP that will be prepared. This is the chance to resolve any unknown variables associated with the original development of the IP, potentially at a third party or at the sponsor. allowing then the compounding pharmacy then to have the ability to do a live demo, to work at scale, and to address any issues that may impact the ultimate batches that are to be dosed.
Absolutely, which is a great segue into the batch compounding is what we're at. And basically this is done only after the results come back from the demo batch. And those demo batch, as we know with sterility, is a minimum of 14 days that needs to be sent out for. The stability in the assay obviously can be determined by the sponsor with the IP product to best understand how they want to move forward on that front. But sterility is something that must be obtained and understood before you go to batch compounding. If the stability in the assays show that the drug is not stable for a period of time, let's say less than 14 days, then what you'll see is that the demo batch can be used as a presentation to the agency that you can produce this IP product under a sterile environment and produce sterile product, and that when you move forward with batch compounding, you're able to say we will run in parallel with batch compounding and dosing along with sterility testing if need be. Otherwise, the demo batch will suffice, provided the data there is to support your position for the batch compounding respectively. This can either be done with an IND that is an open, or it can be done in presenting this along with your IND to the agency, this batch record as well. And when the batch record is first done at the demo batch as well as the batch compounding, the sponsor representative is typically on site at the on-site pharmacy for such compounding to occur to ensure and also see how the compounding is being done and to assure that there is no issues, and also to obviously see what's happening with the bulk IP compounding as it relates and leads to IP dosing.
And I want to add, Harry, the importance or the significance of the sponsor being present during these activities, because typically there are very short timelines between the batch compounding and dosing as usually required for these early studies in order to minimize the need for extensive stability studies, et cetera. The sponsor and plant allows rapid decisions and collaboration to be done in real time to address any unforeseen activities that, any unforeseen items that pop up during the execution of the batch.
Correct. As we look at the final sign here as it relates to IP dosing, this is the test IP which has been bulk compounded, batch specific, protocol specific as it relates to the clinical trial in early phase. This has been IRB approved, subjects have signed an informed consent, and they're ready to dose. We have good documentation from the batch compounding to address sterility, stability, and assay and dose. If not from the batch, from the demo batch, as the sterility is obviously greater than the stability of the drug. That can all be bundled in your presentation to ensure the integrity of the IP product for the clinical study in which the dosing will occur.
Next slide, please, Chris. So thank you, Harry. I want to spend just a few moments for our audience members discussing why we are promoting compounding pharmacy and differentiating that from a traditional good manufacturing practice type provision for clinical supplies. I want to delineate on some level the differences between compounding and manufacturing for our audience members as well, for those that are not familiar with the differences between the two types of approaches. And as our audience members will follow on the slide that's up now, we really have somewhat of a differentiation in the two approaches. As Harry's mentioned early today, most of what is ongoing with regards to compounding investigational product is actually very much in line with what most pharmaceutical sponsors would expect either from an in-house GMP shop or a third-party contract manufacturing organization or a CMO in regards to the types of processes and documentation to support clinical supply material. There are some key differences as we look to the slide. First and foremost, compounding pharmacies do not strictly adhere to the GMP regulations. The GMP regulations themselves, specifically 21 CFR 210 and 211, are exempt or not applicable directly for phase 1 clinical supplies, and there are several guidances that have been put out in that regard. With that being said, as Harry mentioned, the overriding guidance that compounding pharmacies adhere to then are the USP 797 high-risk compounding, or 795 for non-sterile preparations. Most people in the call are familiar with a more traditional approach where you go to a full GMP shop that would either be an in-house manufacturing and testing facility and or a contract manufacturing organization for the third-party provision of investigational product for dosing. You'll also note that the compounding pharmacy has an advantage in the fact that its batch sizes usually are very limited, sometimes on the order of single units for dosing. There's not a requirement to scale to meet minimum equipment designs, etc., to facilitate extreme losses at small scale for traditional compounding or manufacturing, as would typically be seen in a CMO type arrangement. The compounding pharmacy can prepare anything from a single dose up to tens of doses, for instance, to support a single dose PK study to support a phase 1 safety assessment. Lead times are significantly reduced for compounding pharmacies. They typically work on an as-needed basis and understand very well that the products that they are compounding usually have limited stability and limited utility in the clinical setting to support a single clinical study that will be dosed usually within the same or nearby facility. In doing so, the supply's lead time can be on the order of 30 to 60 days, leading up to a fraction of the time or leading up to dosing in clinical studies very rapidly. Take that in contrast for those of you who have had to deal with outsourcing clinical supplies manufacturer to a CMO. Just getting a proposal and a contract in place can be on the order of 30 to 60 days, followed then by another 30 days to procure materials, another 30 days to write batch records, and then to compound or to manufacture the batch, followed then by extensive release testing and releasing the batch. It's not uncommon for a simple phase 1 clinical batch at a CMO to take on the order of 4 to 6 months, and sometimes in extreme cases where there is lead times associated with special components, etc., that those lead times could approach 9 months to a year. For those of you who have worked with CMOs, you will also note that the cost to manufacture phase 1 clinical supplies on a unit dose cost are very expensive. There is no economy of scale at the phase 1 level. The compounding pharmacy, in contrast, can actually be quite reasonable compared to a contract manufacturing organization because of the limited amount of space, personnel, and equipment that they need to manufacture the specific number of doses to be given during that phase 1 study. In all cases, both organizations or both approaches provide the same end result. They produce products that are suitably stable for their intended use. Again, that is depending upon the amount of information that sponsors are able to generate either during the demo batch or through other sort of studies that may be conducted prior to engaging the on-site pharmacy or CMO. Both organizations or both approaches do require some sort of in-process and release testing, as Harry had mentioned previously. It is not uncommon to assure sterility through the demo batch, which would be very similar to, say, a tech transfer batch at a CMO. The finished products intended to be dosing will also be characterized. Depending upon the stability and requirements, they may be characterized either prior to or after dosing. In both cases, it's not uncommon that those products are tested for very similar attributes such as assay-related substances, endotoxins, etc. And in all cases, there are qualified individuals that are assuring that the process, the environment, the training, et cetera, is adequate and suitably documented to support the dosing of the products for the clinical study.
And John, along those lines, talking about USP 797 high risk, we all know there are 3 levels, and we're specifically delineating here this is only a high risk opportunity, number 1. And this bulk IP compounding has been around for years. It was truly mainstreamed in the early 80s, in which bulk compounding in the pharmacies was accepted, and the boards of pharmacies actually have statutes as it relates to this as well.
Very good. And real quick, Harry, I forgot to provide a little more differentiation between how one would view compounding versus manufacturing for our audience members. Most people would expect when they hear the term manufacturing, you're thinking dedicated equipment, you're thinking or multi-use equipment in third-party facilities, producing a batch that is not necessarily study or site specific, that is made in order to support potentially more than one study and supporting distribution potentially throughout the U.S. or even abroad. Contrasting that with compounding investigational product, the batches themselves are very small. They're meant to be used specifically for a single study as described in an investigational new drug application, for instance. The process and the labeling and the utilization is typically done in a very small area. It's not uncommon, like for those services at DaVita, where the compounding pharmacy is co-located with the dosing site. Some organizations will have that across the street or within the same city, for instance. But that the compounded investigational product is study participant specific. It is very phase specific in terms of the dose and say in the dosing in the number or the protocol order, et cetera, and is not intended to supply very large studies across multiple clinical sites, et cetera. Next slide, please. So at this point, there are a few key questions that we hope that the audience has been able to glean from the initial introduction into investigational product compounding by an on-site research pharmacy. First being that pharmacies can compound investigational product for early-stage clinical studies. As Harry had mentioned, the practice really came into favor in the 80s. It has continued to be widely used, especially early in clinical development, mostly, again, focusing on phase 1 type studies or very early phase 2 studies were negotiated. With that being said, there's a long history of use. It has been with it has become mainstream in the media with the advent in 2012 with retail compounding and some of the issues and the issues associated with compounding pharmacies that have kind of gone astray and have become into distribution and kind of blurred the line with manufacturers. And again, that's important to recognize that those media reports are based upon retail pharmacy settings, and there has not really been this out-of-control issue seen with on-site research facilities that compound under very specific protocols, under very elaborate environmental controls and monitoring scenarios that Harry had mentioned that support phase 1, phase 2 clinical studies under an IND. And again, the take-home message that a lot of folks need to appreciate is, yes, the FDA does allow this practice, and it is something that is often negotiated in order to facilitate a very rapid dosing and approval of the IND. So at this point, I want to hand over to Sam for a poll question that we'd like the audience members to participate in.
Thank you, Harry. Thank you, John. So, yeah, it's time for our first poll that we're running in today's session. In a moment, you'll see a polling box appear on the right-hand side of your screen with the polling question and a selection of answers. Please select the answer that's relevant to you, and then remember to click Submit. You'll have 90 seconds to vote. and I'll launch the poll now. And today's question is, how often have you used an IP compounding approach to provide drug product for a clinical study? The answer is A, never, B, 1 to 2 times, C, 3 to 5 times, D, 6 or more times. I just want to take this opportunity to remind all the attendees that you can submit your questions in the Q&A banner, and remember to send them to all panelists. So, Harry and John, I don't know if you just want to comment briefly on why you wanted to ask this question today.
Thank you, Sam. Obviously, we appreciate everyone's time today to participate on this webinar, and we wanted to best understand what the experience is for the people that are currently online with us as we have this discussion. So we appreciate your open, honest candor here as we move forward with the slides and best understand how we can prepare for these in the future as well.
Very good, Harry. Thank you. And just to add, again, I think most people probably have heard of this as in some sort of circles, and I think that as pressures to develop products become greater, that people will begin to will fully appreciate the power of the ability to move in the clinic as quickly as possible using this approach.
Okay, thank you. We've had a good turnout of the attendees voting, and I'll just display the results now. It looks like A, never, has won, nearly 50% of the vote. If you just want to comment briefly on the results and then continue on with your presentation.
Harry?
Thank you, John. I can't say I'm not surprised. We appreciate those that have never been in this position to take the time today to be here for this opportunity to have this discussion. For the other ones that have had the experience, we appreciate you also taking the time to be here today as we have this discussion. The conditions for bulk IP compounding really lies at the heart of the commitment of the compounding pharmacy. And currently right now, legislature is being further developed, but is truly, there's a gap between the FDA and the state boards of pharmacy. And so the onus is on the compounding pharmacy to really step it up and best understand that USP, though it is a guideline, make sure that they are compliant, and not only compliant, they are verified by a third party. What this means is that the compounding pharmacy is taking the extra steps to make sure that they are meeting USP 797 standards for high risk, and they're also working with their boards of pharmacy for the boards of pharmacy to understand what it is that they're doing as a compounding pharmacy as it relates to bulk IP compounding as well. Anybody in a pharmacy position can say we're doing this, and they don't have to take these extra steps. It's the intent of these extra steps to show that they're committed to the level of the integrity of USP, to the level of the integrity of the boards of pharmacy, to adhere to sterile process, class, clean room design, process validation, not only being compliant, but not only bringing in a third party to just verify and validate that they are doing it and doing it correctly. Next slide. The process validation is critical for the compounding pharmacy, for the non-sterile to sterile compounding, as well as John referred to earlier with 795 for non-sterile services. So primarily the discussion today is on non-sterile to sterile with USP 797 high risk. And this basically comes to the experience of the compounding pharmacy, the verification of the compounding pharmacy by the third party, the design of the compounding pharmacy meeting the USP 797 standards, and also making sure that this is an ongoing process that is reproducible and that it's not a one-time verification, that this is ongoing every 6 months to ensure that the compounding pharmacy truly has met the integrity on all fronts. Next slide.
So with that being said, then, we want to spend a little bit of time discussing how a sponsor in collaboration with an on-site research pharmacy progresses from that initial engagement then to the generation of appropriate documentation to support batch compounding. As Harry had mentioned earlier in the presentation, there is an initial discussion where the sponsor will speak with the compounding pharmacy to relay and provide information on the target investigational product to be produced. With that being said, those directions are then reduced to official documentation, typically in a batch record or batch record-like document. That document then is usually circulated to a much larger group of individuals or stakeholders, such as the quality assurance groups within the sponsor or quality representatives with the contracting pharmacy. That then, any comments are then duly noted and negotiated as best to address. And then with that being said, then the final documentation is approved prior to execution. And that is important to note that, again, the expectation for assurance and quality assurance is that there is a negotiated understanding of what will be produced to support the clinical study, and hence the drafting, the reviewing, and the finalization of appropriate batch documentation prior to compounding ensures that all parties understand what is to be done, and that should there be any deviations from those written procedures, that they are duly noted and executed. Next slide, please. I want to talk briefly about a few specific aspects of the batch record. And again, when we say batch record, there's both the initial batch documentation that is used to support the demo batch, which becomes the basis for finalization and generation of the batch documentation to support the actual clinical production of the IP for the clinical study. With that being said, the batch documentation typically includes and identifies all the necessary equipment and supplies that will be used during IP compounding. There's a focus on utilizing unit of use or disposable compounding supplies wherever possible. As Harry had mentioned, these include sterile syringes, disposable needles, transfer tubes, or commercial diluents from various third-party vendors that supply these to hospitals, research pharmacies, etc. In cases where there are unavailable disposables, pre-sterilized components, say beakers or special glassware, the compounding pharmacies that operate under 797 will have access to autoclaves or other sterilizing type equipment so that that equipment can be appropriately sterilized prior to use. And it's also important to note that the demo batch is executed with the same materials, equipment, and process as the intended clinical batch so that there are no surprises moving from the demo batch to the production of the clinical supplies. The batch record also allows an inventory to be taken of all the components moving into and out of the clean room area during batch preparation. It takes into account those items which are used and which are then removed during the compounding as it proceeds. Next slide, please. It's very important from a very early outset that the parties decide on the scale to be used to produce the clinical supplies. In this case, it could be some number of individual doses, some number of volume, etc., so that the records can adequately reflect any overages and process needed in order to ensure delivery of the required amounts of materials. This scale also needs to include allowances not only for dosing, but any sort of in-process testing requirements, retains, etc. It also is important to recognize that another definition for scale is weighing, and weight tracking throughout the batch compounding process is also one of the more well-used in-process controls during compounding pharmacies. It's also important to recognize that we utilize calibrated equipment for everything that we do in this scenario. As Harry had mentioned earlier about going from non-sterile to sterile for high-risk compounding, the typical approach, which you will note, is identification and selection of an appropriate sterilizing filter. The filters used in compounding pharmacies are typically pre-sterilized, unit-of-use disposable. Like a manufacturing organization at manufacturing activity at a CMO, these filters are tested after use to ensure that their filter integrity is maintained during the process. And should they fail their bubble point testing, etc., most batch documentation includes wording or instructions on how to re-sterilize the product using a new filter. Of particular note, for products that are unable to be sterile filtered, there are alternatives such as gamma irradiation, etc., that could be used as an alternative and are evaluated on a case-by-case basis. The batch documentation includes instructions on sampling and how to sample for items such as sterility testing, for any items needed for stability or post-dose testing, as well as any sort of retention samples. One thing that we haven't really discussed to date that I want to include here as well is the generation of labels for the clinical supplies. Compounding pharmacies are in a unique position to provide study-specific labels that are applied to the investigational product prior to dosing as part of the compounding activity. These are very specific to study subjects, numbers, and what's very nice in this regard is that the on-site pharmacies are in a unique position to provide and create single-blinded studies and single-blinded supplies for early clinical studies. Of particular note, it's important to recognize that like a CMO, the compounding pharmacies only compound one product at a time in order to ensure there are no mix-up between actives, placebos, or various strengths of product that may be required for a given study. Next slide, please. So when an individual goes into an on-site pharmacy and begins to evaluate the pharmacy's capability to deliver a compounded investigational product for a phase 1 study, it's not uncommon for an auditor or a technical individual to assess the environmental cleanliness of the compounding area. As Harry had mentioned, USP 797 is very specific about the types of environmental controls and environmental monitoring that must be done. It is also important to note the access of the pharmacy to commercial unit of use pre-sterilized supplies, that these individuals are appropriately trained both through routine aseptic manipulation in the case of high-risk compounding, as well as ongoing process training, batch record training, etc. That these individuals and facility is in a position where that they routinely qualify the compounding process. And again, this goes back to the importance of appropriately executing the demonstration batch. And through that, there's the verification of the supplies, both in the demonstration batch, which can be used to support the process in an IND filing, for instance, as well as verifying that the supplies meet the intended stability, etc., with consideration towards release testing for the actual doses to be administered. And lastly, that there is an appropriate release testing procedure in place, especially for the clinical supplies to be dosed so that each of those dose batches is appropriately characterized going into the clinical study. In this particular case, as Harry mentioned, it's not uncommon that samples from the compounded investigational batch will be sent out to third parties for sterility assay, etc. Next study, please. Next slide, please. Sorry. Again, going back to the demonstration batch, it's used to ensure it's reproducible to the clinical batch. Pharmacists are the individuals primarily responsible for the actual manipulation and compounding of both the demonstration and the clinical batch. The demonstration batch, like the final batches to be administered, are done under a batch record, albeit the batch record for the demo batch may be less specific, as it is more of a tech transfer and an opportunity to identify points of concern for implementing the process at the research pharmacy. Next slide, please. The demonstration batch, in order to support its utility in demonstrating that the on-site pharmacy is capable, is typically fully characterized. This includes assay-related substances, sterility, and others that are dependent on other attributes as depending upon the product that's negotiated with the research pharmacy and the client early on. The demonstration batches mimic and mirror exactly how the investigational product will be compounded prior to dosing for the actual clinical study. And again, it's used to confirm the pharmacy's capability, the personnel training, as well as to ensure that the product to be compounded meets the desired quality characteristics, and that the product is stable within the confines and the environment of the pharmacy to be used. One last note in the demonstration batch, this is also the opportunity that sponsors can use in working with the on-site research pharmacy to do additional testing, such as compatibility testing with IV or intravenous line sets, different types of bag components, etc., as needed to support their IND filing. Next slide, please. It's very important to recognize that a responsible on-site research pharmacy will not compound any investigational product for clinical dosing without the sponsor's approval. And again, that normally comes in the form of approved batch documentation for the clinical investigational product, as well as receiving word that the client or sponsor has been through their IND hold period and that they are free to progress. Again, there are written procedures for the random sampling to support various product quality testing, such as sterility, endotoxin, etc. We have listed here some of the minimum turnaround times for certain types of tests. It's important to recognize that your product stability may fall within or outside of these minimum required testing times, and that how you negotiate the testing of the actual clinical supplies will depend upon the stability of the particular compounded IP. It's important to recognize that the batch record is reviewed both by the sponsor and the compounding pharmacist prior to dosing of the IP in order to ensure that everything has been prepared correctly and that there are no unusual observances or occurrences that need to be addressed prior to dosing. Next slide, please. Thank you. So again, in a brief summary of how one progresses with an on-site pharmacy, as Harry mentioned previously, you negotiate and set up a demo batch, which serves as almost a tech transfer batch that allows the pharmacy to internalize and work up a training procedure for the process to be done. It then allows the individuals to take samples and submit those for various release testing, followed then by generation of the batch documentation to support clinical studies, as well as getting information for the release testing. This then results in product becoming available for dosing in the clinical study. Most of this is self-explanatory in this particular slide. I just want to highlight a couple key items here. One is that the staff is trained, and that training is very important for this type of approach where it's a very manual procedure or very manual compounding process, and that this process is well documented through batch documentation, etc. It's also important to recognize that this operation and preparation of compounding IP is done with a pharmacist and a secondary individual, much like that expected in a GMP setting. Next slide, please. I then want to conclude, prior to going into the last summary slide, a couple recommendations on why we would select one pharmacy over another. One aspect is the pharmacies are compliant with their state laws for operation as a compounding pharmacy. There is typical oversight by a local or state agency as well. Harry had also mentioned third-party organizations that will come in and certify various aspects of the pharmacy, and that they are performing pharmacy functions in accordance with expectations for an operating pharmacy. They have a certification associated with 797 USP for high-risk non-sterile to sterile compounding, and that the pharmacy staff is adequately trained. That training is documented, and that documentation can be provided to the sponsor during an on-site visit. Next slide, please. So Harry, you want to give a few words as we wrap up?
We really appreciate everyone's time today. The opportunity here is to look at your resources, your finances, and your calendar, and to really look at the opportunity of bulk IP compounding in meeting those needs as it relates to minimizing your costs, minimizing the amount of time to produce the IP product, and obviously to ensure the sterility, the stability, and the assay of the drug to be dosed, that is GMP-like, to really come to a decision quickly on appropriate next steps with your IP as it relates to early phase clinical research. Next slide, please. So, bottom line is the pharmacy that provides bulk IP compounding can facilitate flexible dosing by making dosing changes fairly quickly in a matter of weeks rather than months as it relates to moving your product forward, specifically with changes that may need to occur. So we hope this slide presentation from John and myself today has given you a better overview of the opportunities of an on-site compounding pharmacy in meeting your IP compounding needs.
So we'd like to hand it back over to Sam for our second poll question of today.
Thank you very much, Harry and John. So yeah, it's the final poll of today. It's the same as before. In a moment, the polling box will appear on the right-hand side of your screen with the question and answers. Please select the answer that's relevant, then click Submit. You'll have 90 seconds to vote, and I'll launch the poll now. So the question is, would you consider using a bulk IP compounding approach in the future? And the answers are A, yes, B, possibly, but I have additional questions about quality, C, possibly, but I have additional questions about safety, or D, no. If you just both like to comment briefly on this question before we wrap up.
Sure, thank you, Sam. Our interest is in asking the audience today about their approach and would it be considered is hopefully driven by what they have learned today. Knowing that 50% of the audience has not used this approach historically to support an early phase 1 study, hopefully they will glean enough information to entertain the evaluation of on-site research pharmacy compounding of investigational products to support their next clinical study.
Okay, great. I'll just close the poll now and publish the results. They should be available any second. So as you can see, yes has won with 58%. If you just want to comment on the results, and then yeah, I guess we'll go straight into the Q&A section afterwards.
Barry, I don't know about you, but this is a wonderful response. Given that it seems that hopefully our message in terms of the process, in terms of what can be done to support early clinical studies for that 50% of individuals that had not utilized this approach previously to support a clinical study, is supplemented by enough information in today's webinar that they feel comfortable going back to their groups and providing feedback that there is an alternative to the long lead times and costs associated with third-party CMO, GMP manufacturing of clinical supplies for their studies in order to expedite their programs.
Agreed. This is a wonderful response, and it looks like we have several questions that have popped up at this point.
Yeah, great. So as we're slightly short on time, I'll dive straight into the Q&A section. And the first question for you both is, could a sponsor propose this approach as part of a pre-IND meeting?
So Harry, I'll take this as someone who's worked on this side with multiple individuals and companies to propose this approach, and the answer is yes, Sam. It is not uncommon if a sponsor wishes to explore bulk compounding at an on-site research pharmacy for an early clinical study that they propose this approach in a pre-IND meeting with the agency. In this case, normally what ends up happening is the client makes its case for the utilization of the bulk compounded IP, followed then by how they will address and ensure that the product meets the required quality attributes such as assay, sterility, etc., and get the agency's feedback on whether or not there are additional concerns that they have or other procedures that they would like implemented in order to support that early phase 1 study.
Fantastic, thank you. So I'll dive straight into the next question. How many batches should be made for the demo batch to demonstrate reproducibility?
John, I'll let you take that first, and then I'll respond after you for what's physically been done at DaVita Clinical Research for the studies that we've done compounding IP.
Very good, thank you, Harry. So, Sam, that's a wonderful question, actually, and it speaks to how complex the potential process is. If it's a simple solution phase product where you have a soluble compound that's soluble in a standard diluent such as normal saline, and the batch requirements are not overly large, so we're talking about maybe tens of bags or tens of doses, if you will, versus hundreds of vials, for instance. Then in those particular cases, a single demo batch may be suitable to demonstrate that the process is reproducible or performs as expected and can be used to generate sufficient supplies to support release testing or process capability testing through assay, sterility, etc. If it's more complex, then it's highly likely that you may need more than a single batch. Harry, what's your experience?
The experience that we've had here is that I'm going to break it down that there are two primary objectives that usually need to be met. The first one is sterility, and the second one is assay concentration. And when we talk about sterility, that's really complemented by process validation. Does your pharmacy, does your staff have data to support that they have the experience and the reproducibility to go in and produce sterile product through process validation from non-sterile to sterile, and that data needs to be there. If they can do it in the demo batch and that lines up with process validation, that question is answered. As you look at concentration assay, if the demo batch is done and is documented and there were no deviations or no unusual occurrences, then it should be assumed that you should be able to reproduce that along the same lines for batch to batch to batch, and that indeed has been the case. There's only been one occurrence, which I would say less than 2% of the time do we see more than one demo batch being done.
Okay, great. Thank you for your in-depth answers there. The next question is, in view of the meningitis outbreak due to poor air quality in a compounding lab, have rules changed?
You want to speak to that, Harry, or would you like me to take a stab? I'll take the first one. You can follow up. How's that, John?
Great. Wonderful.
Well, obviously, when the 483 came out in October, I think it was 2012, it obviously hit the boards of pharmacy, and it hit the FDA, and it hit the legislature directly. There was existing FDA guidance under 503B specifically addressing research compounding pharmacies and basically meeting the GMP intent and having them register directly with the FDA. That is being beefed up on the FDA front. The boards of pharmacy are beefing up their verbiage as it relates to compounding pharmacies. Neither one of these agencies wants to be held accountable for another New England compounding pharmacy issue. So they are both addressing this. They have draft guidance out, currently under review, with the anticipation of guidance being legislation in 2015, respectively.
And I think it's important to note that the second part of the question was, have the rules changed? And I think fundamentally, Harry, no, the rules haven't changed. It all goes back to whether or not people follow the rules and ensure the necessary quality in their personnel and in their environments all along. And I can't stress enough that it's critical in selecting an on-site research pharmacy that the sponsors engage early and provide an on-site visit in order to assess compliance with expectations such as, and statutes such as USP 797, as well as ensuring that the pharmacy and the pharmacy itself is maintaining its certifications through third-party metrics companies in order to ensure that their biological safety hoods are operating in compliance, that their environmental controls are correct, that their sampling, environmental sampling is current and up to date, and that they are executing what they say they are doing. So it goes back to ensuring that they are compliant with expectation.
Great, thank you very much. I'll crack on with the next question. Are you familiar with the MHRA approach to have industry utilizing X-temp compounding require content analysis before dosing? Do you know if USP/FDA may take a similar approach?
So Harry, if you'd like, I'll take this one.
Go ahead, John.
Okay, so Sam, thank you. This is actually a great question, and I should note that today's presentation is based on application of bulk IP compounding as it relates to phase 1 studies in the U.S. I am familiar with the content of the question requiring the European regulatory agency requiring some testing of compounded product prior to dosing. One of the things that I apologize if it wasn't more clear is that the expectation where possible will be that compounded IP is tested prior to dosing. What ends up happening a lot of times is there's a negotiation between the agency, the on-site research pharmacy, and the sponsor based upon the stability of the compounded IP prior to dosing. If your compounded IP is stable, there is an expectation that you will submit samples from that compounded batch for assay-related substances, sterility, endotoxin testing, much like you would do for releasing a batch at a CMO, for instance. The real benefit here, though, in compounding at an on-site pharmacy is the ability to potentially negotiate post-dose release testing or post-dose testing of the compounded batch where product stability cannot be achieved. The specifics here would be that the sponsor with the on-site pharmacy has a procedure whereby the compounded doses are prepared, say, within 6 hours of dosing. So in this effort, all of the training is done, the batch documentation is completed, and the pharmacy is set up to make those clinical doses in real time in order to minimize any sort of growth potential. So one aspect is recognizing that the environment is in place, the staff is adequately trained, batch documentation. On-site research pharmacies take it one step further by ensuring the minimal amount of time between dose preparation and dosing. But again, specifically to the question, where possible, the batch samples from the batch would ideally be tested prior to dosing.
Okay, thank you very much. What looks like our final question today is, please provide more information on COA from sponsor. Is this for the API?
I'll start first, John, and then you want to follow up?
Go ahead. Thanks, Harry.
Okay. Typically, the COA that you would receive from the sponsor with IV product, we normally are aware of that documentation. When we're doing bulk IP compounding and we have ingredients that will be added, the product may come from the sponsor with a COA for the non-sterile product, or it may not. If it does not, then we will actually do the physical compounding, and whereby it's sent out for sterility to one venue, it's sent out for stability possibly to a second venue, and sent out for assay development to a third venue. With these three separate parties, that data must be collected, in which at that time the documentation can be pulled together for COA of the compounded IP product respectively, which is then placed into the batch record and tied back to the dispensing product. And I know we're short on time, so John, do you want to add to that?
That's correct, Terry. So I just want to, again, aligning that with expectation that that the misconception about high-risk compounding in an off-site research pharmacy is not a controlled process. Hopefully through today's webinar, the attendees appreciate the GMP-like approach to on-site compounding of investigational product. And with that includes the testing of the investigational batch, as you had mentioned, Terry, which is typically done both in-house through certain tests that you have as an on-site research pharmacy, but also using third-party laboratories that are more specialized in in HPLC or other analytical techniques. And that data is then collated and is then put together into something that resembles a certificate of analysis for the batch, and those results are then assembled along with the batch preparation documentation to provide a complete batch history of the characterization and preparation and characterization of the investigational product.
Okay, great. So thank you to the attendees for your questions. Thank you, Harry and John, for answering. If you just want to give any final thoughts briefly before I wrap up today's webinar.
I personally would like to thank everyone for their time and for the opportunity to have this discussion as it relates to a new paradigm for opportunities or options for compounding IP product. And should you have any questions or wish to have additional follow-up information, we have an email on the screen there for kevin.grudeau@devita.com, or if you wish to call Kevin directly, he will be your primary contact, in which he can then filter and make sure that the information is disseminated appropriately, and we could respond appropriately to your request.
Thank you, Harry. I just want to reiterate my thanks for everyone's participation today. I hope that everyone will be able to walk away with something new from today's webinar, especially the idea that there is an alternative to traditional GMP third-party manufacturing of clinical supplies to support early-phase clinical studies, that this type of on-site research pharmacy compounding is an acceptable practice within the U.S., and that it does have its merits in facilitating time to first dosing in man, for instance, in early-phase clinical studies, as well as exploring some of the early work while minimizing investments in drug product dosage form development during early clinical work.
Okay, fantastic. That just leaves me to say thank you, Harry and John, for what was a great presentation. Thank you to DeVita Clinical Research for sponsoring this session. The presentation slides and a video recording will be available on demand on our website, which is www.business-review-webinars.com shortly. We look forward to sharing further webinars with you, so please keep an eye on our website, as just mentioned. Follow us on Twitter @BRWebinars for daily updates, and join our LinkedIn group, Business Review Webinars. So thank you once again, and I hope you all have a nice day.
Thank you.
Thank you, Sam. Thank you, everyone.